Good afternoon, everyone. Today we will discuss about translation or protein synthesis. So prior discussing about the main topic, we have to discuss, rather recapitulate the previous one, that is central dogma. In my last class, I have discussed about central dogma, that is, when I say that in our college there are two different types of libraries. One is the departmental library where no students are allowed other than botany, and second one is central library, that is where all the students have free access to that library, and that is why that library is known as central library. And likewise, all the living organisms continue or perform a common pathway that is known as central pathway. Before discussing the central dogma, we have to discuss about the biological macromolecules or biomolecules. There are four different types of biomolecules, each with their respective function, like the carbohydrate, which is responsible for the production of energy in the form of ATP. Next, protein, which have constructive power, lipid, which is the storage molecule, and the last one is nucleic acid, which perform or which function as the genetic material. Genetic material is that material which restore the characters of the organism and transfer the characters from one organism to the next organism. And the main function in the central dogma, like the transcription, where the mRNA or rather RNA is formed from the DNA, and translation, where the amino acids are aggregated together by means of peptide bond and form a polypeptide. The main function performed by the genetic material. We know that there are two different types of genetic material on the basis of their pentose sugar. One is the DNA, where the pentose sugar is deoxyribose, and the second one is RNA, where the pentose sugar is ribose. Actually, ribose is a parent molecule and deoxyribose is derivatives of ribose, and that is why RNA is primitive than the DNA. Well, the organism which have both the DNA and RNA, DNA function the genetic role. Only the organisms or living entity which have no DNA, rather possess only RNA. In that case, only RNA perform the genetic role, like some RNA viruses. Now, what is central dogma? Quite better to say, living organisms are mortal in nature. Mortal in nature, that is, they have a certain lifetime. It may be twinkle of an eye to millions of years. And in the second part, living organisms try to maintain their copy in this universe. That is, in one part, they are mortal in nature, and in the other stage, they are trying to maintain their sustainability. So this contrasting character is maintained by a biological phenomenon, what is known as reproduction, and which is mediated by the cell division. And reproduction is the production of an organism where new individuals are produced from a previous existing organism. In central dogma, within the cell, the nuclear material or the genetic material is present in the nucleus, and there outside the nucleus there is cytoplasm, where different types of cytoplasmic organelle and cytoplasmic materials are produced or present. In central dogma, first DNA replicates itself to produce two copies of DNA within the nucleus, and the protein synthesis is performed in the cytoplasm. Now, from the DNA, one copy of RNA is produced within the nucleus, what is known as transcription, and from this, the RNA are of three different types depending upon their function. Number one is messenger RNA, second one is tRNA or transfer RNA, and third one is ribosomal RNA. Messenger RNA carries the message of DNA, and according to the codon of mRNA, amino acids are invited from the amino acid pool, and the amino acids are joined together by means of peptide bond or CN bond and produce a polymer of amino acid, what is known as polypeptide. So the DNA replication, where the template is DNA and the product is new DNA, is carried out by the enzyme DNA polymerase. Actually, it is named DNA dependent DNA polymerase. Usually we call it DNA polymerase. In the next step, using DNA as template, RNA is formed. Both are the polymer of nucleotide, and the process is known as transcription, and it is carried out by the enzyme RNA polymerase. The actual name of the enzyme is DNA dependent RNA polymerase, but we usually call it as RNA polymerase. And from the RNA, the amino acids are joined together and produce polypeptide or protein, which is primary structure of protein, and this process is known as translation. Why it is called translation? We know that RNA is the polymer of nucleotide and proteins are the polymer of amino acid. In our lower class, there is some Bengali sentence and we have to convert it in English, and usually we called it translation. And likewise, in this case, the nucleotide sequence, that is the RNA, is converted to the amino acid sequence, that is the protein, and that is why this process is known as translation. And translation is performed in the cytoplasm. Next, what are the requirements of translation? We know that translation is performed in the cytoplasm. It requires the mRNA. mRNA stands for messenger RNA, which carries the message of DNA from the nucleus to the site of protein synthesis, that is cytoplasm. And protein synthesis is usually performed on the RER. RER stands for ribosomal rough endoplasmic reticulum. Rough endoplasmic reticulum is the endoplasmic reticulum which carry ribosome on their surface. Next, the requirement is ribosomes. Ribosomes are nothing but it is a nucleoprotein component. The nucleic acid, that is rRNA, is responsible for the production of ribosome, and the ribosomes are associated with mRNA, what is known as polyribosome, and according to the three nucleotides, we will discuss next please, three different types of RNA, that is mRNA, messenger RNA, which carries the message of DNA from the nucleus to the site of protein synthesis, that is cytoplasm. Next, tRNA, it stands for transfer RNA. Transfer RNA, transfer the amino acids from the amino acid pool. Amino acid pool is located on the cytoplasm where different types of amino acids are gathered, and in tRNA transfer the required amino acid from the amino acid pool to the site of protein synthesis. And third one is the rRNA, which is known as ribosomal RNA. Ribosomal RNA construct the ribosome. Next, how a protein is produced? A number of amino acids. The instruction for making a protein, a series of 3 nucleotides on the mRNA, what is known as genetic code. Genetic code, mRNA is produced using the DNA as the template. We know that in nucleic acid, the nucleotides are joined together in a complementary way by hydrogen bond. So, the sequence of DNA in the nucleotide in the DNA is complementary to the sequence of nucleotide in the mRNA. And in the mRNA, 3 nucleotide is responsible for accepting one amino acid, and this is known as codon. And as this codon, that is the 3 nucleotide, is produced by the genetic material that is known as DNA, and for this reason this codon is known as genetic code. Codon signifies 3 different types of codons. One is the start codon. Start codon, usually start codon is AUG, which codes for methionine, and this is the starting point of protein synthesis. Second one is the stop codon. Stop codon, I think stop codon, there are 3 different types of stop codon, UAA, UAG and UGA, and it signifies the stop of the protein synthesis, and the remaining codons specifies one amino acid. Next, please. This is the calendar of genetic code. Yes. Anticodon. That this is the structure of a tRNA, what is known as cloverleaf model. Here there is 3 nitrogen base or nucleotide, which is the anticodon. This anticodon interact with the codon of mRNA, and this transfer RNA carries one amino acid, and this is known as amino acyl tRNA. Next. This is the ribosome. These are the all requirements. Ribosome. In prokaryote, the ribosomes are of 70S type and consisting of 2 subunits, 50S and 30S. But in eukaryote, the ribosomes are 80S, consisting of 2 subunits. One is the larger one is 60S and the smaller one is 40S. Next. And there are different types of the ribosomes mRNA start codon. Start codon is usually AUG, and it codes for methionine. But in some cases the start codon may be GUG, but usually in the middle portion GUG signifies valine, but where the GUG is function as the start codon, it always signifies the methionine. Next. This is the polypeptide chain. So next, the protein synthesis. What are the different types of protein synthesis proper? Protein synthesis proper consists of 3 different steps. So translation include number 1, requirements for protein synthesis. Second one is the activation of amino acid, so that the amino acid can be transferred from the site, amino acid pool to the site of protein synthesis mediated by the tRNA. And third one is protein synthesis proper, where the amino acids are jointed together by bench of peptide bond or CN bond and produce the polypeptide chain. And this protein synthesis proper consists of 3 different steps. Number 1 is the initiation, number 2 is elongation, and third one is the termination.